on the screen. If you are given a specimen, what are the steps that you would follow to classify it? So the question is asking, if we are provided with a specimen, then how or what are the basic steps to classify that organism into its respective category? So first of all, what we are going to do, we know that there are some fundamental features. There are various kind of common fundamental features we have in the classification system of animals, such as what type of symmetry is present in them, what type of coelom they are exhibiting, what type of symmetry they are exhibiting, what type of organization is present in them, if or whether the coelom body cavity is present in them or not. So these are the basic fundamental features, fundamental characters on which or with the help of which we actually can classify the different organisms into their respective categories. All right. So what we are going to do, if we are provided with a specimen, the steps would be follows as, first of all, what we are going to do, the answer would be like this. There are various common fundamental features by which we classify the given specimen into its respective category. So what are going to be these categories? First, we'll consider the level of organization. Now, we know that we have two types of level of organization, or three maybe. So, if we talk about the level of organization, the first phylum of animal kingdom, that is Porifera, phylum Porifera, including all the sponges, are exhibiting cellular level of organization. So, first is we have cellular level of organization. Cellular level of organization, which is found in the primitive animals, most primitive animals of the animal kingdom, that is sponges, which are belonging to the very first phylum of animal kingdom, that is phylum Porifera. Then next, if we talk about, we have tissue level of organization. Tissue level of organization. Also in this we have organ level of organization. Organ level of organization. And the last in this we have organ system. Organ system level of organization. Alright. Now, this is the number one feature on which we can decide or put the given specimen into its respective category. Now, also we have type of symmetry. Type of symmetry. The specimen is actually exhibiting. So, we have two types of symmetry basically, that is radial symmetry as well as bilateral symmetry. And if any organism is not exhibiting any kind of symmetry, then it is known as to be asymmetrical organism. Now next common fundamental feature we want to take to classify the given specimen into its respective category would be the presence of coelom. Coelom. Now what is coelom? You all know that a fluid-filled cavity is present. We have a fluid-filled cavity present in our body between the body wall and a gut wall. If any animal is having a fluid-filled cavity between the body wall and a gut wall, then the animal is said to be coelomate animal, because there is a presence of true cavity which is filled with the fluid. That cavity must be present between your body wall and a gut wall. That cavity is filled with the fluid and is known as coelomic cavity. That cavity is known as coelomic cavity, and if any animal is having that cavity is known as coelomate animal. If any animal is having cavity but the cavity is not lined by mesoderm, then the cavity is said to be pseudocoelomic cavity, and the animal exhibiting or having pseudocoelomic cavity is known as pseudocoelomate, as in Aschelminthes. If any animal is having no cavity between the body wall and gut wall, then they are known to be acoelomate animals. For example, Platyhelminthes. And we are the example of coelomate animals because we have a true cavity between our body wall and gut wall. So coelom, in the coelom we have coelomate animals. Coelomate animals. Next we have is pseudocoelomate. Pseudocoelomate. And the last one is acoelomate, in which there is no cavity present in between their wall, body wall and gut wall. So this is the complete answer. This is the complete solution for question number 2 of animal kingdom.